All right. Damn. Yo, Jason, come take a look at this. Get these things into super focus. Look at those.
Oh yeah, you're just high focus. Look at one. That's not like that.
You gotta increase it again. Go another one. See, because I'm on the third one, yeah.
Guys, just write your name on that lab packet that I gave you, because I'm just gonna ask to like look at it quickly before you guys leave, so I know who's here. But then I will collect it, I'll give it back to you guys so you can do your assignment. All right, maybe we'll find myself. Did you get it in focus by everything? Yeah? What do you got? So try to switch it one more up to the floor. Yeah? Let's see, you might have to focus it a little bit to, you know what I mean, to get it in there. And then I'll pull it up on it. You guys don't have to take a look for it. All right.
Hey, bro, I want to say no. Like, you can't see the nucleus.
Yeah, I can.
Because I can't.
Wait, actually?
Yeah, I don't think you
of.
can.
Kind
Right, you don't see the nucleus, right? Yeah.
No.
It's not strong enough power. So, no.
What the hell is this? You can see it because it's not strong enough to see, right?
Mm-hmm.
Starch solution on the tray there, right in front of you guys. Do you?
No.
I didn't miss it, right? It'll do. All right. This. You guys have a note? All right, you guys can kind of share. I'm just going to put one of these petri dishes per table, and I'll tell you guys what you're going to do with it. You guys can kind of share that. You know what? I'm going to leave it over here. Never mind. Because then we're going to have to transport this around. So we're going to leave it here. So we can kind of do this in groups, and I'll explain that in a second. Let me just check one more place to see for starch.
Yes.
Did you get it in focus under high power?
Yeah.
Yeah? Nope. All right, whatever. Okay. Missing one thing that I'm looking for. Right? All right, so what we're doing, guys, right? You guys, hopefully everyone made the slide, right? So basically you have a leaf, right? An aquatic plant sitting in water, okay? So for plant cells, they like to be in a slightly hypotonic environment, right? So a plant cell is gonna be healthy in water, okay? So if you guys look at the picture that's on your, your first page there, let me turn down the light in the middle here. I'll turn these guys back on. So I'm just gonna pull that picture up on the screen too, so that you guys can see what I'm talking about. And let me move, this is so annoying. Because I probably need to turn, you know what, let me turn on, I'll turn this guy on. Whatever, I'll figure it out. Okay. I can just easily. Okay. So if you guys look, just so you guys see up here. So if you guys look up here, you guys that are on Zoom, you can just look at your lab packet on the first page. So this is what they're doing. They're basically putting this LED leaf under the microscope in water, okay? And so the only thing that you really can see with the microscope here is the entire cell, right? Or a couple cells. But if you look within those plant cells, and if you look up here, all those little things that you're seeing inside are chloroplasts. So chloroplasts are pretty big, right? So that's what you can see. You really can't see any of the other organelles, okay? Does that make sense? So what you're seeing there are the chloroplasts. And that's kind of what we're going to pay attention to. So the plant cell, right, plant cells that you see in that microscope are happy right now. The chloroplasts are all evenly distributed throughout the cell, okay? So it does ask you in number 6. So I want you guys to answer those questions that are below there. So can you locate the cell nucleus? Now, look, under high power, you can kind of look carefully. You might be able to see a nucleus. However, because there's so many chloroplasts, it's kind of hard to see sometimes. And it kind of looks just like a, sort of like a darker kind of shaded region.
Yeah, like
okay?
a big shade.
So you might be able to see it. So just see if you can find it. And then, you know, it's kind of asking if you can find it or not. So like I said, it's kind of like shaded, okay? And then again, let's answer those questions that are there. So you guys that are on Zoom, remember, I'm going to ask you to email me this PDF that you're working on. You can handwrite it. You can do it in, I guess handwrite it is what I'd prefer. Or handwrite it in Cami, don't print it, because it's very easy to copy that way. And I'm gonna ask you guys to email that to me, okay? So when lab's over, you guys need to email me this PDF. But that's not the assignment. The assignment is to do the mini lab report. You guys don't need to do anything except show me, right, when you're done, okay? All right, so what about number 7? Why can't you see the other organelles, you think? Why don't you think you can see like, I don't know.
They're too small and the microscope's not strong enough.
Yeah, they're too small. So that's the reason why, right? That's it, pretty much it. That's why we see the chloroplasts. They're larger, they're easy to see. Like I said, you should be able to see the nucleus, but just because there's so many chloroplasts, it just makes it difficult to see. That's all. Okay, now look in there again and look to see if you think that the chloroplasts are moving. Okay, and then I'll take a peek in some of your guys' microscopes just to see. So you should be under high power now, under 40x. If you need help getting in focus, let me know, okay? Yeah, I can help you, yeah. That's all, like I said, I wanted you guys to get started and then I can help you guys. Okay, yeah, so first things first, whenever you want to get it in focus, right, you want to start at the lowest objective. Okay, so that's the 4 here. And then you'll just switch up to higher and higher. So right now, I want you to do, you just want to basically see that leaf in there. It's going to be obviously a little bit larger, but you're going to use these here to adjust the focus. Okay? Yeah. Oh, is your light on?
Hey, bro, did you sign up for the weight room yet?
No, not yet. I'm waiting a few weeks.
I'm going today.
Yeah, I'll be your
I'm tired. I was tired of waiting, bro.
Yeah, me too. It's just
I don't think that there's, you don't see a light.
No, dude, there was so many open spots until like 2 o'clock.
The
bro.
microscope
I
guy
literally
is coming
signed
to
up
fix our
like
microscope. So.
a half hour ago, right before I came here.
I should have.
I was like, I got nothing
You
else
guys
to
see
do.
chloroplasts moving? do. You probably see some movement. It's not because the chloroplasts themselves move, but do you know why it looks like they're moving? What do you think moves around inside the cell?
Everything else?
Yeah, I mean, like the cytoplasm is fluid, right? So you can see things kind of moving around just because the cytoplasm moving. All right. So here, get this on. I'll put it up here and then turn that light on.
Are they at the edge?
Get it in focus.
Are they just chilling then?
Yeah.
No.
Slide it all the way in the corner, and then attach it to that. Okay? Now to move it around.
I mean, everything's like basically what you're looking at. It's already moving, but
Yeah. Okay? So now see if you can get it focused. All right, let's see here.
I think that was the point of the question, that everything's always moving.
Yeah, so yours is all right. You can kind of see down there.
I know we'll see it in a few. When we do those, we'll see him go. We'll see him come in.
Yeah, no, that'll be. Yeah, so that looks good. So like kind of like towards the bottom right there, you can see there's a whole bunch of chloroplasts inside those cells.
Cool.
You agree? You kind of see those spots kind of evenly distributed through. All right, so if you guys have that in focus, you saw that. I want you to, and I'll come back over to help you over there, don't worry. So if you have that in focus, right, we answered those questions. It says save your slides for later, but let's do that part now. So now take that slide off, flip to page 52, okay? You're gonna make a new slide. Instead of a drop of water, you're gonna put a drop of sodium chloride, okay?
So we're taking a new leaf, right?
You should have sodium chloride. You can leave that, keep that.
So we take a new leaf then?
Yeah, take a new one. Make a whole new slide with a new leaf, and there's sodium chloride here. So a drop of sodium chloride instead, okay?
So
Get a
I
new
got
leaf,
you.
new slide, new everything. All
Sodium
right,
chloride.
we have it here. So, okay. All right, so you got it kind of in focus, almost, right? So now you just want to turn this a little bit more.
I want a big one. Pause.
All right, now look
Yeah,
back.
bro, a little
Now it's like
session.
completely in focus now? Okay, all right, cool. So now, we're gonna switch to the higher power. So now this is gonna magnify it more, right? All right, so now look in there and then just adjust this little knob here so it looks clear to you.
This one, right? Uh-oh.
Did you get it?
What you going for?
She said sodium chloride.
Yeah.
How do you open it?
Yeah. Did you do it on the same one?
Like a cork?
Okay. Yeah, so now same
Twist.
thing. Put it on the slide and drop sodium chloride and flip it over to it. So instead of water, guys, you're putting sodium chloride on it now, yeah? You got it? All right, cool. Let's see. Yeah, okay, good. Now, we're gonna go one more. So now, switch it to go to
So
the
one
blue
drop,
one,
right?
right?
Yeah.
Very good. I'm trying to click in. Yep. Same thing. Then use this little knob to adjust it
Two
to
drops,
get it
it's okay.
in focus, right? So it's clear. Once you guys get, same thing, get that in focus under 40x again, right? Under high power. And then there's a table at the bottom. You just basically want to write down what your cells look like in terms of where the chloroplasts were when you had it in water and the hypotonic, and then when you have it in sodium chloride, right? So we're kind of contrasting what they look like. All right, let's see. All right, good. Yeah, so that looks good. So you see all, so now it's not going to be completely in focus, right? Because it's kind of like a little bit of thickness to it. But you can see all those green spots in there. Those are the chloroplasts, and you can see that they're kind of, well, they're kind of like that picture up there, spread out, okay? Yeah, so now you want to answer these questions here, right? So, see if you can can find the nucleus, you may have to kind of like move it around or adjust this a little bit. All right, yeah, so now the table. So now basically, guys, in that table, you're comparing what it looks like with the sodium chloride versus the water before, okay? Then it's asking you, due to what scientific term? So for that part, it's referring to, where's those terms? See if I've written anywhere in here. I'll help you. Oh, it's referring to up at the top paragraph on page 52, turgor pressure or plasmolysis. Okay? So again, when it's in hypotonic, that's the water. Hypertonic is the sodium chloride. Okay? What do they look like? Plasmolysis or turgor pressure? There we go. Did you find it, Liz? Maybe not. Okay. So then just put it
52.
down. That's fine. And then it says, why can't you see the other organelles? So the only thing you see are the chloroplasts, because the other
Bless
organelles.
you.
Thank you.
Right? All right. How you guys doing over here?
Chilling.
Does it look different now?
Yeah.
Maybe. No, it should. You should see all the chloroplasts kind of squished into the center now of the cell. You guys get it in focus? So it's never, you'll still have, it's still like a little, all right, what do you got here? Use it in focus here? Okay, yeah. Then you gotta go under higher now. So now get it in focus here. You put it in sodium chloride?
Yeah.
Okay. So look on this end because that's, I see where the liquid is.
Yeah.
Come here, I'll get it in there for you. Yeah. All right. All right, now you wanna go one more to high power and get in focus and look in there, okay? You're good?
Yeah.
You guys are good on Zoom? Does anyone have any questions? So again, we're on page 52 now. And we're going to complete that table based on what we saw before when it was in hypotonic, and then what we see now when it's in a hypertonic environment in the sodium chloride. All right, so now you're going to make another slide. Yep, you got it, just like you did with that one, except now put a drop of the sodium chloride.
What are you saying?
Jar. So put one drop of sodium chloride, and then color. Yep. Good? Yep. So does it look like that picture that you see in the lab manual? Maybe? Or your lab handout? Did you notice, like, could you tell that it was kind of shrinking to the center?
Well, what is she? What's she
Yeah,
asking
okay.
us, like?
All right, so let's complete that table under there. What'd you guys write so far?
For hypertonic, they shrink to the center of the cell.
Correct. So for table 4.5, right? For hypertonic, they shrink to the center of the cell, right? And you can see that picture right above that table, okay? And then what is that? What do we call that in terms of the scientific term?
Plasmolysis.
You ready? Did you guys focus? Make sure, all right, yeah, let me help you. Let's make sure, let's see where the little power.
That's for hypertonic, right?
Yes, hyper, correct, is the sodium chloride, and that's when they shrink to the middle.
It is called plasmolysis, right?
Mm-hmm.
Is this your sodium chloride one? Yeah. Alright. So you may have to kind of adjust the focus a little bit to get it perfect for you, but I think you should be able to see now, yeah? All right, so we're gonna do this. Good, perfect. All right, so now, well, you may have to back off to get it in focus, but it may be pretty close. Go the other way. Yeah, one more. Yeah, get it in focus first there, then switch up to the high. What do you got here? What did you do for this table? This one? Yeah, so right, you're right. So shrink to the center, good.
What do you for hypotonic?
Yep. Okay. Yeah.
Chloroplasts are all over the
Just
cell.
maybe shrink to the center.
They're not specified to like the walls or to the middle. They're
Get
kind
it
of just
under high power
moving
again.
around. Because that's what we saw when we were
I'll
looking
come
at
back,
this,
make sure
the first
everyone's
slide. The chloroplasts were
good?
in the middle and
We
they
got
weren't
this.
at
All right,
the
so
edge.
what are
They
you
were
guys gonna
just
write
showing
for the
all over.
hypotonic? That's what we did first. So in terms of where the chloroplasts were, that's what they mean by the appearance of the cells. What do you think? What'd you write?
For what, hypotonic?
Yeah, for hypotonic. The first one we did for the water,
I
for
just
the
said-
appearance, what'd you say?
I just said that the chloroplast, you can kind of see them all over the cell. They're not
Okay.
really specified to the wall or to the middle.
Yeah, so they're kind of spread out throughout the cell, okay? All right, and then, and you kind of do see them. I mean, they are kind of spread out. Yeah, I guess they are kind of spread out all over. They do have the tendency to kind of be pushed more towards the cell wall and the plasma membrane, because when plant cells have a lot of water, the central vacuole is filled, and that takes up a lot of space and kind of pushes things towards the outside, but whatever. Spread throughout is good enough. So what's the scientific term for hypotonic? Would you guys write down? Turgor pressure, right? So again, it's because that central vacuole is filled with water, it kind of pushes against the cell wall, and it gives it that rigidity, right? So it's like the difference between like a withered plant that has no water versus like a strong sort of structured plant, right? That's well watered, okay? All right, so make sure you guys got all that. Make sure these, well, if anyone's there, just make sure they know what's going on. If not, no worries.
If you guys get confused, just ask. I'll help. I don't know.
Okay, so now go one more.
So as a teacher for the day.
Are you guys done? Yeah.
I mean, I don't know what they're watching. They're watching me fucking curly.
And then I'll tell you guys what we're gonna do next. The next part we can kind of do as a group or in two groups, okay? You don't all have to do individually. This I wanted you to do individually just because we're using the microscope again. Full practice. Got it? Since you guys are finished with this part, you can just put your slides, just sit them here. And then you guys can turn off your microscopes and unplug them and cover them and just leave them right where they are though. You lost it? Look, it just had it.
Oh, yeah.
Okay. Yeah, so if you kind of look at these ones that are kind of in the middle, you can see how they're kind of all, you see how it kind of squishes in kind of to the middle of the cell, so you can kind of get outlined. And so some of them, right, not all of them. All right, cool. So now we're gonna fill this part out. Then I'll get everyone, we'll be back together again. So
Or you can play grass.
the first
You
one.
could like play grass, like by the grass rules. That's just one stuff.
What, today?
No, like whenever we end up playing,
Oh,
like
fact.
you get the win because that's just fun.
That's a fact.
Write that down. And then what
You think we're gonna start
scientific term
soon?
goes along with that? When
You think
plant
so?
cells are in a
We can get out to the last
hypotonic
one.
solution, if you look up at the top here,
I think we're gonna start.
it's either one of the two. All right, so the next thing we're gonna do, guys, just to, if you guys wanna kind of start looking at this, we're gonna do the diffusion, the speed of diffusion. So that starts off on page 48. All right, so so just read, no, I lied. It starts on page 47. Okay, so just start reading through that, okay? All right, so now for the hypertonic, right, what did it look like? In terms of where those chloroplasts are. You can even look at the picture that's in your handout there too, right? Yeah, but so go by kind of like these, like what it looks like, that it kind of squished into towards the middle, right? Because that's kind of what I'm trying to focus on. And then just write down what that corresponding scientific term is. All right, so for those of you guys on Zoom, we're moving on to, well, we're kind of going back, but we're moving on to
page 47, speed of diffusion, okay? And this is what your little mini lab report is going to be on. All right. So yeah, so just take a look at that. So now we're going backwards. Sorry, we're backwards. Now this is what we're doing. Yeah, we'll do this together, okay? You can shut this off. Just put your slide on there, cover it. Let me move this side. All right, so let's see how we're going to work this out. Like I said, we'll kind of do this in groups. You kind of have to. So for the air one, we usually use, and that's the last one, so we can do that last, but we usually use an air freshener. So one person kind of stands here. I think we usually count 10. Yeah, 10 squares. I'll tell you what the distance is. So one person has a timer. You can use your phone, right? They spray and they start the timer. And then the other person down there is going to tell the timer when they smell that air freshener. Now, obviously, we got mask on, so it's going to be a little bit, I don't know, different. But I think we'll still be able to smell it. So we might as well just try it. Who cares? And we'll have a number there. So basically, you're going to record the times for all of these things in this table here. Okay? All right, so that's, but we can go in order. I just wanted to kind of let you know about that. Like I said, we'll probably just do that air one together. Plus, if you spray multiple air fresheners, you start smelling the one over there instead of this one. So we'll do it together. But everything for this speed of diffusion, you're recording in this table, okay? And then, like I said, your assignment, if you look on Moodle, is to do a little mini lab report, which essentially you're going to put this table, remake this table in a nice, neat way, put that in there, and basically you're answering these 2 questions that are there, okay? And then you're also answering the lab review questions, okay, which are on page 56. So that's your assignment in a Word document. You're just putting the data from the speed of diffusion, right, together with these lab review questions, okay? So just so you guys know what you're doing with that. It's not that bad. It's pretty simple. Okay, so let's look back on page 47. The first one is solute diffusion through a semi-solid, okay? That's already set up for you. So basically what we're gonna do is we're gonna come over here, okay? We can kind of do this one at a time, or one person can kind of do it and tell the group what it is. But you're gonna measure from the center. So you'll see this is just basically an agar plate with a hole in the middle. So we basically put some dye in the middle, okay? So what we want to measure is from the middle kind of that circle, that hollow circle there, to the edge. So it's, what is that? That's almost like the radius of the circle, I guess, okay? And that will be the distance that it diffused, okay? So basically you're writing that down. Oh, well, you have, so sorry. If you're still on page 47, you don't have to go to the table just yet, right? It says obtain time 0 from your instructor and record time 0 and the final time now in table 4.2. So time 0 was 8:35 a.m. Okay, so right? So for semi-solid, time 0 is 8:35 a.m. Okay? Time 0 is 8:35 a.m. And then what time is it now? Oh, perfect. Let's go with 3:35. 3:35 p.m. is the final time. Got it? And then you want to write in the length of time in hours next to it. So we don't have hours and minutes. We just have hours, right? So length of time in hours. What's that? 7 hours? Okay. So we got 7 hours.
Over here, like, is it?
9:35, 10:35, 11:35, 12. So yeah, all right. So that's in the semi-solid column. Time 0, 8:35. Final time 3:35. Length of time is 7. Okay. All right. So who wants to go measure how far it traveled in millimeters? Because that's what you're going to add in next. So here's a ruler. There you go. Somebody measure.
I can't read, man.
Who's closest?
Let's go, Jason.
Come on, Jason.
Oh
you can
man,
measure.
I'm gonna struggle.
we can help you out. So millimeters, right? Remember those are the tiny little lines? So you kind of want to estimate and get this part in the middle of that hollow circle and then out to the edge. That's all. Tell us how many millimeters it is. He can do it, I think. I mean, you can add it. Wait, little lines, not the big ones. The little tiny lines. What is it, 15? Okay, cool, 15 millimeters. All right, your job's done. Well, for now, right? Okay, so now we added in our semi-solid column, distance moved is 15 millimeters. Okay, so now it next asks you to calculate the speed of diffusion. So how are you gonna do that? It's millimeters per hour. So how do we calculate the speed of diffusion in semi-solid? What's your math that you're doing there?
Divide 15 by
Super
7.
easy, yes. 15 by what?
7.
Correct. So we're gonna do 15 millimeters divided by 7 and write that down in your table. Should do long division.
2.15.
I was just been doing that with my daughter. That's horrible. I haven't done that in like years. I don't even understand.
141.
Anyway,
Was it around?
142.
what'd you get?
Okay.
Yeah, so it's-
Yeah, something like that, right? Like 2.4?
yeah, 2.14.
2.14? Okay, so 2.14 millimeters per hour. Make sure you put the units, okay? All right, so that was easy, right? All right, now the next one is liquid. So same thing. Somebody can set this up for all of us. So what you're gonna do, I guess I'll just, we'll bring it over here. You guys might have to walk over here. Just keep your distance, though. All right, honey, you can set this up. So just pour some water. Just pour some water enough to fill kind of the bottom, or maybe like halfway up, kind of. Oh, wait. Put your, we're gonna put the ruler underneath, okay? Yep, fill the bottom with water. Good, pour it. Yep. All right, let me move my computer. They're wondering what's going on. All right, so here we go. So now we're doing the liquid, yeah? Okay. Oh, do we need a times 0? No, I don't think it. Oh, yeah, okay. So for this, times 0 is 0, doesn't really matter. And then, or you can just put 342 and then it's gonna be 352. Okay, so times 0 could be 342, final time is 352, because they want us to leave it for 10 minutes. Although I'll tell you that's gonna be too long, but whatever, who cares, we'll go with it. 352, so the time is gonna be 10 minutes, okay? In that table, yeah, you guys see that? Okay, it does ask you though to convert 10 minutes into hours. So remember in your table, it has to be in hours. So somebody convert 10 minutes to hours. What do you do? How do you convert 10 minutes to hours? Well, how many minutes are in an hour? 60. Yeah. Yeah, what? Is it right? So we're trying to convert minutes to hours. So what do we just do? Divide by what? Right? Yeah. Divide by 60. So what is it? 1 divided by 6? 60. 1 divided by 6? What?
0.6.
Yeah. Right. All right, so get that in hours so we have it all set up properly. So then you guys don't really have to do much when you go home to do your lab report because we're doing it together now. All right. Now, what we're gonna do, so we have this all ready to go, okay? So now Hannah's gonna put some dye right in the middle of this liquid, okay? So you just wanna try to put it like right here at the 0 mark, right? And then we're gonna see what distance it travels in 10 minutes.
Maybe not.
Yeah, you just do the best you can. Like put a little tiny bit in the middle there. Yep. All right, and then we're gonna have to try to do our, I gotta go make the air pressure. We'll try that.
I think I know what I did.
We'll call our distance, yeah, 3 meters, I think. That's what I usually do.
Oh, that's it?
Yeah,
So it should be 0.16.
yeah, that's what I was gonna say, yeah, I think it's 0.16. Definitely not 0.6, right? Because that would be like 40 minutes. Right, 0.16 of an hour is your time, guys. Yeah, see, so you're gonna see, so we'll be able to stop this early, but, all right, so we're gonna leave that right now, okay? And in 10 minutes, or around 10 minutes, you guys are gonna we'll remind each other to just measure that, okay? Yeah? All right, so that's set up for liquid. So for the liquid, we had 342 to 352. The length of time in hours was what? 0.16 hour, okay? And then we will get the millimeters, okay? And then you'll calculate the speed again, yeah? Okay, all right, so for air, let me go get the air freshener. We'll try it out. Huh?
What scent is it?
Oh, I forget, let's go see. It's probably some gross scent, but whatever. Be right back. It's dried sage, fig, and brown sugar. Okay?
Sounds solid.
Yeah. Awesome. All right. So for this, okay. So now we're on the air. So let's see. What does it ask us? What's at the top here? All right. So again, yeah, you're gonna have to calculate the time, right? Essentially. So like I said, somebody's gonna stand here with their cell phone and their timer ready. As soon as they spray, they'll hit start, okay? The other person over here is gonna say, okay, I smell it. So whenever you smell it, you're gonna let us know, and then the timer will hit stop, okay? Yeah? So we'll know the distance. So the distance, we're gonna call this 3 meters. Meters. So you gotta convert that into millimeters, I believe. Right? In the table, it says, it does have millimeters, right?
Yeah.
So for air right now, we can set it up as 3 meters is what? How many millimeters? Uh-oh, how many millimeters are in a meter? How many? 1,000, right? So what, 3,000? Okay, so 3,000 millimeters. All right. Okay, and then so like I said, we're gonna get the length of time. That's what we're determining, okay? And then we can get our speed. All right, so who wants to do this? Take your own cell phone. You guys wanna do it? Somebody with a cell phone, huh? You're not gonna really put anything, you're just gonna put length of time because you're gonna hit time 0, you're gonna hit start, and then we're just gonna get that. So there's not really a time 0 and a final time, okay? All right, so somebody with their cell phone and somebody with the air freshener. One person is here, and then 1, 2, 3, 4. The other right here. All right, cool. Right there. You got it.
So I take the spray.
Yep. So as soon as you spray,
When I smell it?
correct. When you smell it, you say stop, and as soon as you spray, you hit start.
Okay.
You got it. Ooh, that's a big spray. All right. Good. Now let's see. This is probably gonna be a little different because we got masks on, but I'm sure we'll smell it.
I want to smell it, but it's so bad.
Can you smell it?
No.
Shoot, man.
I don't really smell it either. I guess our masks work, huh?
Good thing.
How about this? You can pull your mask away just a little to see if you smell it.
Yeah, I smell it.
All right. Did you smell it yet?
Yeah.
All right, stop.
Smells like some laundry, like.
It's fig, brown sugar, and sage. All right, 33 seconds. All right, so for our air, right, the final time, the time zero and the final time, you could just put a line through, that doesn't matter. But the length of time is 33 seconds. Oh my goodness, how are we gonna get that into hours? Oh my God, I think we can do it. We can go in.
It's 0.0091.
What'd you do?
We had to get it into hours, right? So
Yeah.
I just divided 33 by 60 and then that by 60.
And then by 60 again, right? Yeah. So what is it? Point what?
0.0091 or 92 because it rounds.
0.0092
Yep.
hours. All right. All right, so now you can calculate the speed of diffusion if you want to, right? Because now we're just dividing our 3,000 by 0.0092. So obviously you're going to get a big number for this one, right? What'd you get? 326,086. Okay.
Yeah, put a decimal.
No, you don't need the decimal. So it's, what was it, 326,086? What's the units? Millimeters per hour, right? Yeah?
Yeah.
All right, so what do we get? Let's review. For semi-solid, what was our speed? 2.14 millimeters per hour. Okay. All right, what about liquid?
We should be collecting
Oh,
that in a
read
minute.
it now. Give us the distance. Yeah, it's close enough. It's already the edge anyway, I'm telling you. Right? Yeah, so you want, right, what is it? 30 millimeters. So for our liquid, the distance moved is 30 millimeters. Moving this back over here too. Well, I guess not. They can see what we did here. A little closer. All right, so for liquid, we did 30 millimeters. We already knew our hours. It was what, 0.16?
Yeah.
Okay, so 30 divided by 0.16, yeah? So what was it for liquid, the speed? 187.5 millimeters per hour. Yeah, we got that, guys? Everyone got that here? You good? Yeah, okay. All right, and then for air, it was this, right? 326,086 millimeters per hour. Okay, so then if you look at the questions underneath, it says, in which experiment was diffusion the fastest? Air. All right, so why? That's what the next question is essentially asking you. So why do you think diffusion was fastest in air versus liquid versus semi-solid? We're going to put for a reason there. And that should be, those two questions will be in your little mini lab report, so make sure it's there. So what do you think? Brad?
Doesn't it necessarily have to do with the amount of freedom the atoms have?
Yeah, I think that's a good way to put it, right? That there's really nothing obstructing their movement, right? And so they are more free to move, right? So they're going to be able to move faster. We're going to add to that? All right. Cool. So we all got that? Makes sense? When we're talking about diffusion, what is diffusion? Let's just review. What is it? Right, but when you say, so think about your air freshener, right? And we think about diffusion of those odor molecules essentially, right? There was a high concentration here, right? In which way did they move? in the direction of the low concentration, right? So when we think about diffusion and everything we just did, we had a concentration of dye molecules in one area, and then we measured how fast they moved out to the area where it was less concentrated, right? So whenever we're thinking about diffusion, it's just really the movement of the molecules from high concentration to low. That's it, okay? And we just demonstrated it in a few different ways, okay? All right, so like I said, for your assignment, you basically are just making a nice table like we just, with all the data that we just put in here. Okay, we did the assignment already, right? Answering these two questions, the only thing you have to add is the answer to the review questions, okay? So in your Word document, it's that table, the two questions, and then the answers on page 56, okay? We have one more thing to do, and then if you guys want to sit and do the questions together, you can, or if you want to do them, and if you have a question for me, I'll hang out. That's up to you, okay? Go ahead. Correct. It's these two questions, and then flip all the way to the back. Those questions.
Do you want us to put 33 seconds underneath the total time, like times 0 and the final time in the air column, or do you want us to just leave it blank?
You can just leave the in the air column time 0 and final time blank.
Okay.
Okay.
Can you repeat that?
For what? For
We're due to the mini.
yeah. So for the mini lab report, right? It's basically you're remaking this table in Word in like a nice table, right? With all the data we already put in there. You guys have everything you need to put in there. Answers to those two questions that we just went over. And then it is the answers to the lab review questions. Okay? Got it? So that's, you'll see the assignments on Moodle. It says mini lab report. That's all you guys are doing. Okay? It's pretty simple. Again, we pretty much did almost the whole assignment already. All right. So let's just do one last thing. So the last thing we're going to do, guys, is page 48. Okay, so right underneath the speed of diffusion. We're basically going to set this up, this solute diffusion across a plasma membrane. And again, we don't all have to do it, but you can if you want. So take a look at this and kind of read what you got going on. You have in water the dialysis tubing, okay? You guys can share. We already have some here. So if you guys, why don't you guys kind of start setting it up? If, like I said, if you each want to set it up, you can, but we could just do it as a class again together. It's fun, okay? So what we're first going to have to do is we're going to have to take that tubing out. Kind of feels like a plastic bag, right? And we're going to have to, well, tie off one end. Okay, you're going to tie off one end, and then you have to be able to try to open up the other end so that we can put liquid inside, right? So see if you can get one end tied off and the other end open, okay? And then you'll need another empty beaker to set this up. Here, then you'll need this, Brad, to set that up, okay guys?
Okay.
Jason, set that up. If you guys don't want to do it, just come over here and watch them, okay? All right, so yeah, you can take it with your hands. If you want to put gloves on, you can, but it's honestly, it's like a plastic bag. All right, yeah, here, just tell them what you're doing. Because I'm recording anyway, so then at least it's good. Now let me get these ones.
What the fuck is this thing?
My starch solution.
Your coffee today, man? You like?
Hey, bro, I don't have steady hands. You know this.
I'm gonna get some water boiling, guys, because eventually we have to do a better mix test. All right, so you're gonna tie off one end and open one end.
You want to try it?
I got it.
Oh, you
It's.
got it, you got it.
Get it? It's hard to open.
I have the freaking corner lips.
It is. It's like it's thin. Yeah, you kind of can. If you kind of rub together the end, you usually can get it open. Or maybe like one of those plastic droppers. If you can kind of stick it in there and kind of get it started, that might help too. Let me go see if I can find some more starch. Otherwise, we're gonna have to use what I got there, but it's not an ideal one.
This is so tedious. Yo, I got an idea. Grab your tweezers.
Here, I can help you guys if you need it.
Improvise.
This is the hardest part, is
Pull
to
it.
get this stupid thing open.
Grab the other side. Just pinch it.
There you go. Oh, I had it. Nope.
I'm gonna see that.
What the fuck?
I got it.
Where? Oh, it's open. Finally.
Can't get out.
Don't close you. Good looks.
Got it?
Yeah. No, you. Oh, there we go.
What are we putting in?
I gotta tie it first.
Did you guys get it? Alright, let me help you.
Will it rip?
Oh, and then for the sheep blood, the new blood didn't come in, so I'll show you guys pictures and we'll just talk about it, okay? That's the only other thing.
Blood.
How'd you guys get it?
I
Did you
kind
use
of,
the dropper?
yeah, I used the droppers, the tweezers, kind of just
Okay.
used everything.
Now you gotta tie off the bottom there,
Okay.
okay? You want me to try, guys? Yeah. Right here, we're still gonna have to tie off this one then too.
Alright, we're chilling. What are we filling it up with, the iodine?
Yeah, look to see what it says. I think is it glucose solution you're filling it up first with? Fill the bags halfway.
Oh yeah, it's the glucose and starch.
Alright, cool.
You wanna get the glucose, Jason?
Yeah.
Alright, you wanna get the pipette? I'll hold it open, you fill it up.
Alright, so to fill it, you
You
guys
just
can
rip it
use these
open?
bigger.
Yeah, just pull the,
pipettes.
it's
So
just,
to kind of fill this, I
one of
think
those wax
halfway
covers.
glucose.
Condom.
Yeah, literally.
And then, you know what, we're gonna end up, I don't think you need, wait, is it what you put water in there first? I think so. Yeah. You'll end up needing more water in your beaker, right? 2/3 of water.
You got that
It
hole?
won't, once you start adding the liquid, it's fine. It won't.
Use the big one, my G.
Which one? Oh, yes.
All right, you got glucose, yeah?
Yeah,
So fill it, what, about halfway it says with glucose, right?
yeah.
Okay, halfway. And then I have some starch for you guys. White ice,
Is that halfway?
starch, I'm gonna.
About halfway. I think we're chilling, right?
It's a little more than halfway. Yeah, we're good.
No, that's halfway, bro. We're
Oh,
chilling,
yeah.
my
Oh,
G.
yeah.
I need more water. You wanna fill this up?
Yes. What is this? Distilled water?
Yeah, basically.
How much do you have to fill it?
Just a good amount, to like 100, 150. That's chill.
Yeah, and then make sure your beaker itself is 2/3 full of water.
Okay, we have
this
to
good
add
enough?
starch to this too, right?
Correct.
So
I'm
we're
gonna
just,
get you starch.
all right, we're just waiting on
Yep.
you.
How much stuff do you have?
All right, here's some starch. I think it only tells you to add what, like some drops, right? Not a lot.
Yeah,
4 droppers of
Of starch.
starch.
Small one.
Yeah, use the bigger dropper.
This one?
Oh yeah, you guys need starch too? 1 second, they're almost done.
you can take it, we're good. Oh, he's got.
You gotta
tons
do 4 of those.
of- Okay.
All right, right,
What,
4
what happened
dropper
there?
full of starch, and then
that's
what do you need to do next? You might be able to do that.
Right, I mean,
Droppers of iodine solution to water. Okay, so then you can add iodine here. So just take a new, you should have the thing of iodine there. A new dropper until it looks yellow. You want your water on the outside to look pretty like dark yellow.
fill up that with iodine. Does that look?
So yeah, you can either use your dropper or kind of pour it in. Just use your dropper actually. All right, yeah, so that's fine. And then you're gonna end up
That
kind of
good
placing
enough or
it.
more?
Yeah, do a little
Like
more.
on the angle, so it's chilling, but
Yeah,
the
you
top
kind
is
of like
at-
have this hanging over or
just
whatever.
like that.
Correct. And then the rubber band sometimes can help hold it in place.
Okay.
The outside, not the inside. So goes in here. Yep. Starch goes inside and then this.
Get the rubber
You
band.
probably can put that whole thing in there. Do one more.
Good looks.
You want to have enough iodine there, otherwise it like doesn't work as well. Yeah, here.
Yeah, it's kinda not in
Totally
there.
yeah. The other thing you can do is you can just add a little more water and a little more iodine.
Do you want us to?
Yeah, add a little, yeah. Nah, just more iodine. Add more iodine though. Yep, you guys are gonna add the starch to there and then that's it, right? And then, yeah, make sure the outside like has enough iodine in there that it looks like dark yellow. If it's like super light, just add more. Like I said, it just works better when there's enough iodine in there.
Yeah, I think when we mix it, it'll be better. For the rest of the night.
All right, and then just, if you could, Jason and Brad, just show up to the camera what it looks like at the start and describe what you did. Like I said, it's recorded, so I just want to make sure it's there. Yeah, what do you do? You put starch in there?
Where are your
Or
teachers
dropper
for
full of
today?
that
Yeah,
starch.
so we just
You
took the
wanna-
dialysis bag right here.
Thank
I
you.
don't, yeah, it's kind of in a bad spot. Let me turn it this way. Right there. Filled it up with starch and glucose, and there's iodine in the water around it. Now we're just gonna wait.
We chilling.
Let me do this.
That's enough then. You want to at least have a little bit to hang over. All right, you're good. Now
Could you
you're
close
gonna
up the
put it in
iodine?
here, right? And then you kind of break this over, and then your rubber band kind of holds it in place. Nah, it's fine. Yep, you guys got it. All right, cool. So I think it tells you, how long does it tell you technically it is? Yeah, it says that after about 5 minutes. So we'll talk about something else while we wait for this. If it goes longer than 5 minutes, it also doesn't matter.
Where's
Okay,
my
so we don't like
paper
to, I don't like to waste time. You guys have to figure that out. So,
Yeah, where did you put your paper?
all right, so that looks good. You know what, guys? Put a little more iodine in there. See how dark theirs are? They're just, it just makes it more noticeable when at the end of the
Remember-
experiment.
Oh,
All right, let me pull up our sheet flood pictures just because the new sheet flood didn't come in, so it's not gonna work if I try to use the old sheet flood, so we'll just talk about it.
you guys are comparing. Damn,
All right,
yours is-
so
yours is dark as hell.
before we move on, guys, let's talk about kind of what you guys did here. So you put, what's inside the bag? Let's remember what's inside the bag. So what'd you put inside?
Glucose and
Glucose?
starch.
And starch inside. And then on the outside in the beaker, what do we got?
Water and
Iodine.
Water and iodine. Okay. So now you gotta think back to our lab from last week when we did like the chemical tests, right?
Mhm.
For the presence of starch and glucose, right? So what is iodine? What do we use iodine to detect the presence of?
Starch.
Starch. Okay. So if this dialysis bag is simulating a plasma membrane, what we want to see after the 5 minutes or 10 minutes is what was able to cross,
Mhm.
right? So if starch is able to cross through that plasma membrane, what will happen at the end of the experiment?
The iodine will react.
Okay, so where will you see a color change?
The brown.
Say louder. Yeah, but where? In the beaker, right, itself, right? Yeah, so right, you'll see black or blue kind of in the beaker. Okay, so that's one potential thing that could happen. right now, we don't have anything that's gonna test for glucose, but you guys also have glucose in there, right? So we will, if you guys look, at the end, we'll do a Benedict's test to see if glucose was able to move across, right? So basically what you'll do is you'll take a sample from the water outside here and see if it tests positive for glucose, right? And if it does, that tells you what?
That glucose can go through the membrane?
That glucose was able to cross through the membrane, okay? Okay, and then there's one other thing. You have iodine sitting in the beaker, right? It's possible that the iodine can actually move from the outside into the dialysis bag. And so what would indicate that that happened?
The bag changes color?
Yeah, the bag will change color. You'll see it change blue-black. Okay? So those are kind of a couple possibilities that might happen. For this, the way that things are moving across that is just purely based on size, right? So you can kind of think about that starch, big molecule, right? Starch is made up of a whole bunch of little glucoses, right? So unlikely, if we're talking about size, the largest molecule of starch probably not going to be able to cross. Iodine, just one single atom or whatever, it's just iodine, right? It probably may be able to pass, okay? So we'll see. So basically, whatever happens, that will determine what crossed, okay? So let's leave that sitting. Like I said, all I'm going to do is I'm going to pull up the pictures, if you guys look. At the sheep blood pictures, what the heck page is that? Page 51. You're not gonna have to record anything in this table, but we'll just talk about it, okay? So if you guys look at tube 1, 2, and 3, that's what was set up, and I'll show you the pictures to show you what it looks like. But basically what this is demonstrating is these possible scenarios happening to the red blood cells, right? And again, this is in hypertonic, isotonic, or hypotonic, okay? So if you guys look though at tube 1, okay, it says 0.9% sodium chloride. That's what the sheep blood cells are in. So do you think that tube 1 is isotonic, hypertonic, or hypotonic?
Iso.
Okay, so why'd you pick isotonic? You're right.
Oh, because of the picture? Right?
Yeah, well, yeah, you're right. But tube 1 doesn't necessarily correspond to A.
I thought.
But maybe it kind of does. But so anyway, 0.9% sodium chloride is like physiological saline, right? So that's isotonic to your red blood cells. So that's similar to your body's environment, right? If you look at tube 2, now it has 10% sodium chloride. So what do you think? Is that hypertonic or hypotonic? So more sodium chloride, right? More solute in the outside.
Hyper, right?
Hypertonic, right? And so when you place red blood cells in hypertonic, if you look at picture B, we don't even have to pull anything up, we can just do it right here. If you look at picture B, what's happening to that red blood cell?
Shrinking.
It's shrinking, okay? We call the scientific term for that is crenation, just at the top there, because I think I may use some of this vocabulary in the lab quiz, okay? So when the red blood cell shrivels up, it's called crenation. What was the name for when the plant cell shriveled up? What was the scientific term we used for that?
Plasmolysis.
Plasmolysis, right? All right. If you look at when it's in hypotonic, and that's tube 3. So now tube 3 is essentially like you could have just put it in straight water, right? And that would be hypotonic. So this is basically like a little more diluted saline, okay? What happens to the red blood cell in the hypotonic?
Filled up, swelled.
Yeah, it's swelling, right? Because why? Water is moving in. Remember, this all is a result of water either moving out or moving in, okay? So let me just quickly just pull up the tubes of the sheep blood when you look at this kind of as a whole sheep blood, rather than looking under the microscope at an individual cell. And then we'll go back and we'll look at what happened. Is anything happening yet?
Not really.
Maybe. No? It doesn't look like your tubing looks a little blue.
Nah,
No.
not really.
No?
Either that or we colorblind.
Either that or I'm bugging, but
No, we don't.
nah, it's
That's what's supposed to happen. We'll see. Maybe. I don't know. Whatever. It doesn't really matter.
pour the whole thing of iodine in there.
Word.
Something will happen.
That'll really happen.
Let me just pull up those pictures real quick, and then, like I said, we'll look to see what happened we'll do our little Benedict's test, and then we're finished. All right, so where are we?
Does she have my paper? I don't know.
I don't know. Ask her. Cause
Dr. Kaminsky, you have my paper, right?
What'd you say?
You have my paper?
Oh, I thought I picked your paper.
No, it's okay. No, it's
Sorry.
it's okay.
I forever like leave stuff around and take other people's papers. Yeah, that's nice.
bro, you were like, what?
Yeah.
You're like, what the heck? I took it from the garbage. Sorry. All right, so sheep like
If she did go to
pictures.
disrespect.
If you guys take a look. I'll just
Damn,
tell you
bro.
what they are.
I was like, where'd it go?
Funny as hell. Did you need anything or did you get it all?
No, I need some.
We didn't write anything
So if you
down.
guys
please.
look
So
here,
which one do we write?
right, so basically this is full sheep blood in isotonic, right?
No, we
What's
didn't write
number two?
No, it'll be that.
We'll be saying hypertonic and then hypotonic. So it's basically when they shrivel up, they kind of sink to the bottom. You kind of have a little bit of, I mean, it's kind of like looks clear red. In number three, they've all burst. And so you can see how like much darker it is just because all those red blood cells burst. In number one, it sort of looks sort of normal, like normal blood, okay? Honestly, it's better to kind of understand what's happening at those individual cells
Wait,
that
that's
we talked
the-
about
Yeah.
rather
the
than
blood?
here, but this is kind of what it would look
Do you want
like.
to show them this or?
Yeah, you can, I guess.
That's the blood you're talking about?
Yeah, so that's the sheep blood, and it's on Moodle. There's sheep blood pictures. So thank you. One corresponds to tube 1 in the lab, right? 2 corresponds to tube 2, and 3 corresponds to tube 3.
You want us to write that?
You don't have to worry about writing anything down there. We're going over it. But so, and I believe, yeah, on the lab quiz, if I use a picture, I won't use a picture like this. I'll show you guys a picture of this, right? And say, hey, what solution was that red blood cell in? Was it hypertonic, hypotonic, based on what's happening? Okay. So it's better, I'd rather you guys just look at those individual cells rather than that. Because sometimes it's actually, this looks pretty clear, but sometimes when you do the experiment, it doesn't look as good as this, okay? All right, so like I said, for, let's take a look at these now. I guess we gotta look back at our experiment here. Nothing? So what starts to happen usually is, so let's take a look at this. Right, so what does start to happen, like I said, iodine's small, right? So what you'll see, and I'll just leave this sitting here like until Thursday, and I'll show you guys. But you'll start to see the inside of the tubing gets blue-black because the iodine really ends up being the only thing that can move across, okay? But let's do a Benedict's test real quick. So I gotta get, oh, yeah. So somebody just take some out. You're just gonna take a little bit, I think it tells you to take like a milliliter or something out. So close to your bag, just take some liquid out, squirt it into here. And then we're gonna just add some Benedict solution to it. How much does it tell you to take out? Like a mil, right? Yeah. Okay, so just take it from, suck up a dropper full, squirt it into here. We'll add some Benedicts to it and put it in the boiling water and see what happens. Okay, might as well finish it. I don't know why that, I think it just needs more time. Oh, here, you want to do yours too. Then bring your test tube over here, guys, and you can add the Benedicts to it. You don't need a lot. Just add like a dropper full of that too. Yeah, that's good. And then just be careful. I'll pull it out for you guys, okay? Just carefully place it there without burning yourself. Just add a dropper full of this.
Oh, yours is blue.
Yeah, you gotta put that in it.
I'll put it in there. Okay. All right, we can just let it go for like a minute or so and then we'll check it out. So this is us testing to see if the glucose moved out of the tubing, right, to the beaker, meaning could it cross that membrane, okay? So like for this experiment, important to kind of understand like the setup, right? I mean, not that like you have to like memorize anything. I guess I'll give you like a scenario and kind of, say it was set up this way or something like that. But understand that the dialysis bag represented a plasma membrane, right? And that things can cross based on size, essentially, right? Smaller things cross, bigger things don't. That's pretty much the sort of gist of it. So like I said, we'll give it a minute, and then I'll just turn that off. We don't even need to reach in there to grab anything. We can see if it changes color, okay? And that's pretty much it. Let me just show me your lab real quick so I can write down everyone's names. So I know who's here. And that's pretty much it.
So it's just those two things for the new lab.
Correct. For the mini lab, yep, just the table, those 2 questions, and the review
And
questions.
when is that due? By today?
I think I have it due by Friday.
Okay.
Yeah, so best to just go and do it, right, and just be done with it.
And I'm gonna, you wanna stay in front of these posters?
We're good to go?
Yep. We'll just wait like 30 seconds. Let's see what happens with this.
Cause I mean, like,
No, I saw it. Yes. Just want to make sure I got everyone. Thank you. Yeah, take that because we need that data for the mini lab.
thank you. What do you want us to do with these?
So like I said, if you want to go over any or do the questions.
Just put them on the meeting.
or
Yeah, they're
anything
going
else
online,
here,
and then-
otherwise-
Just to make.
And then,
You
yeah,
guys got it.
along with the draft.
It looks like no color change, guys. So for the Benedict's test, it's negative, so that tells us that the glucose wasn't able to cross,
Did
okay?
we throw out the?
Just leave those there, just leave them.
Okay.
I'm gonna leave these sitting. Like I said, probably we'll see over time, it may turn blue-black. At least that's what usually happens.
We're lucky. You
Oh,
wanna stay? Or do you
Bye,
wanna go
guys.
over?
Thanks, guys.
It's up to you, bro. If you wanna stay and do them, you go to my room.
Yeah, if you guys
and
wanted to do it, you
Yeah,
can.
we can just do it.
It's up to you.
I'm down to do them here.
If you have a question, I can answer it.
Um.
Take care.